Hi everyone, my name is Nancy Auyeung and I'm a PhD candidate in the Molecular Biosciences Department and in Dr. Kurt Horvath's lab. So let me tell you, I'm fascinated about viruses and that our human body has the potential to fight these viruses. And this fascination is what led me to do science research to this day. And what I'm trying to do in my research is understand how does the human body respond when it's infected by a virus. And I'm trying to do that by looking within the human cell and looking at the DNA level specifically, because the potential to fight a virus lies in your DNA. So there's many different viruses that attack on the human system. Some you've heard of, such as measles, influenza. Others you may not have heard of, such as Nipah virus, and many more. But they all affect the human body, which is why I want to understand how does our human body, how does the human cell respond when it's infected by the virus? And I told you I want to look at it specifically at the DNA level. So where is our DNA? So within the cell, there's a compartment called the nucleus. That houses our DNA, the blueprint to everything that our human body can make. And it's being represented up here by the X-shaped, which is the compacted version of our DNA because we have so much material it wouldn't fit if it wasn't compacted. But before I take you too deep into the space of the nucleus, I want to take you back in time, specifically back to 1957. So in 1957, scientists learned that a cell that's been infected by a virus, it secretes a molecule called interferon. Before 1957, and even in 1957, very few people had even heard of the word interferon. But by 1986, interferon was already being tested in human patients that were infected with hepatitis C virus, which causes liver damage. And to this day, interferon remains one of the most effective therapies to treat patients with hepatitis C virus. So now, what happens after interferon is secreted? Well, it turns out it can actually bind to neighboring cells, ones that are uninfected by the virus yet. So why would it do that? Well, picture this. Imagine your neighbor's house was on fire. Wouldn't it be nice if you can receive a signal that alerts you that your neighbor's house is on fire, such as looking out the window and seeing some smoke? Well, that's what interferon is. It's the signal that alerts neighboring cells that have not been infected yet that, hey, there's a virus in your vicinity. You need to prepare. You need to build a defense system to fight it. So that's great. And since 1957, scientists all around the world have found out even more now. What happens after interferon binds that cell? Well, it turns out after interferon binds, it activates lots of processes, and it continues relaying this message that there's a virus in the system and that you need to prepare by building your defense system. And that message gets relayed into the nucleus because that's where the DNA blueprint is, and that's how we can make anything, including the defense system that we want to build to fight the virus. And so this is where I come in. I am looking deep inside the nucleus, looking closely at the DNA and how it plays a role in building the defense system. So let me take you deep inside the nucleus now, and let us focus in on one X-shaped DNA E. So now inside the nucleus, at the top you'll see the blue X-shaped. That's the compacted version of our DNA, because I told you there's so much material, if we didn't compact it up, it wouldn't fit in the nucleus. So now let's imagine pulling it apart so we can see the different layers that might have gone into that compaction. So we might see different layers here, such as the multi-rows of beads as we travel down from the compacted form. We might reach a single row of beads now, and where you can start seeing that each bead is being wrapped around by the DNA piece about 2 times. And now if we look at one individual bead and we start unraveling it some more, we might reach a single DNA strand. So we don't know about all of the levels very well in terms of this compaction process, but we do know at the single bead, this is what it looks like in your cell. Your DNA could potentially be wrapped around a bead, but this isn't just a bead. It's a representation of actually 8 histone proteins. And when I refer to proteins in our cell, I'm not talking about the proteins from your protein shakes or the chicken that you eat, but these are proteins that have functional roles. They perform tasks such as helping fight the virus. So looking at this schematic here where the DNA is being wrapped around the 8 histone proteins, that's great of a method for the cell to try to keep your DNA compact Otherwise, it would just be a big mess and it would be all over the place, wouldn't fit. So, but then it becomes a problem for the cell now. What if it wants to use that information, that DNA blueprint, to make something, and in this case, to build a defense system? That becomes a problem. And imagine this, if the DNA was a ribbon, and on one side of the ribbon was a message, now I take that message that's on one side of the ribbon, wrap it around a ball. That message is now concealed by being on the inside. And that's the problem that the cell could potentially be running into. So if we look along the DNA, different stretches of DNA encode for different genes. So at the top, that might encode for a gene for eye color. And on your bottom right, that could be encoding for a gene for your blood type. And what I'm interested in is looking at DNA segments that code for genes for different antiviral proteins. So there are hundreds of different antiviral proteins. And that's what makes up the defense system that will ultimately fight the virus. So when I started my research, one of the questions I had was, if we looked at a DNA segment that's encoding for an antiviral protein, X for instance, or Y, what does it look like? Is it this free accessible strand, or is it wrapped around and potentially inaccessible for the cell to use, or some variation in between? And that becomes important because if the cell wants to access it, in the case of a virus infecting it and interferon sending the signal into it, what processes and what players come into play? What does the cell employ in order to make that DNA become accessible? And this all goes back to the fact that I believe that studying the DNA, it's one part of trying to understand the human system. How does the human respond when a virus infects it? And like the story of interferon, in 1957, the discoveries and the research that we do today can help fight the viruses of tomorrow. And that's why I believe in science research. Thank you.